Next up, we have proteins. So this is another type of polymer that is made up of repeating subunits or monomers. Proteins are extremely important. They pretty much do just about everything that you can imagine. So we have up here as part of their function that they provide support. So they can be support structures. They can be used for storage. They can store energy. They can be toxins. They can be antibodies, which help to fight off infections, and enzymes. So we'll talk about enzymes later on. But enzymes are types of proteins, and they are important for helping to move chemical reactions along. So such things such as elastin and keratin, which you find in your skin, in your hair, and your nails, these are proteins. Proteins are so critical and so important, and they are some of the most complex types of molecules that we're going to find. And that's simply because they have unique shapes, and their shape helps them to function properly. So in biology, we use the phrase that structure determines function. So the structure of the molecule determines how it functions. And it's important to keep that shape in order for it to function properly. If it loses its shape, then it's not going to be able to work. So we said proteins are polymers, so they are made from a monomer that's called an amino acid. And you've got the 20 in parentheses to show that there are 20 different amino acids. And those are the building blocks. So we can take amino acid, join it to another amino acid, join it to another one. And that string of amino acids is then folded into a unique shape that makes up a protein. And it's within that string of amino acids, and each amino acid has slightly different properties, let's say. So one amino acid might be acidic, another one might be slightly more basic, all right? Some might have charges on it that would make it hydrophilic. Other areas where you've got different amino acids might make the molecule more hydrophobic. So the amino acids are going to help determine how that protein functions and where those amino acids are located. Okay, so it says it's a polypeptide chain folded into a unique shape. So let's look at those shapes. So here we're showing an amino acid. So we're starting out with that building block of an amino acid. And notice for the first time we're seeing nitrogen here. So nitrogen is an important component of an amino acid. The R does not refer to a specific element. This just refers to the fact that most of the amino acid looks like this, and then that R is what we call a side group. So this is where different atoms can be attached, and it causes the amino acid to have slightly different properties. All right, so it's just kind of showing you a generic amino acid at this point with not any specific atoms attached in this area of the molecule. So this amino acid, if we can look at it here and say kind of this orange circle, right, now represents this amino acid, and we're attaching it to another amino acid. Slightly different shade there to represent that there could be slightly different properties between this amino acid and the one after it. Then we attach another amino acid. So they kind of look like a pearl necklace, I guess. These kind of beads that we're making here, that we're kind of stringing them along. And this is what we call level 1. It's really what we call a polypeptide chain, because it's just a chain of amino acids. Then we take that chain and we start to fold it up. So this is level 2, where we can create different coils like a spring, or we start to fold that chain back upon itself. Then as we move on to level 3, at this point we're starting to form different types of bonds, and we're creating, as it says, more of a 3-dimensional shape. All right, so we're kind of folding it out and coiling it around itself to create more of a ball-like structure. And then level 4, we can have several of these, let's say level 3s, joined together. So this could be one right here, another one right here, another one right here, and another one right here, and so forth. So we're creating kind of subunits that are all joining together to make this level 4 protein. So you can see, once again, that this is an extremely complex structure. And one end of it could be more hydrophilic, another end could be more hydrophobic, and that's going to determine how different molecules interact with this protein. All right, so that was a short introduction to proteins, but let's talk about nucleic acids. So nucleic acids are another type of polymer, and we're starting out with our examples of nucleic acids first. So these are two very important examples of DNA and RNA, and you need to know both of them. We're going to spend a lot more time talking about DNA and RNA later on in the semester. So it's really important to get to know these molecules now. So DNA stands for deoxyribonucleic acid. So if you're ever asked which type of polymer DNA is, it tells you right in the name, it's a nucleic acid. So the N and the A refer to a nucleic acid. So DNA carries our genetic information. So encoded in the DNA molecule, encoded in the structure of it, are the instructions on how to make thousands of different types of proteins. All right, so that's all encoded within areas that we call genes. So genes carry the instructions on how to make different types of proteins, and we'll talk about that later on in the semester. Our next molecule is RNA. So the N and the A refer to nucleic acid. So in this case, it's ribonucleic acid instead of deoxyribonucleic acid. So RNA is what's going to help to build the proteins. So it's going to carry the instructions from the DNA out into the cytoplasm to meet up with ribosomes so that they can build proteins. So RNA, that's what I just described as what's called a messenger RNA. There's also what's called transfer RNA that are also important in helping to build proteins. So RNA is a little bit more of a temporary structure than DNA. So we're going to see differences in how they are formed. So let's talk about the structure of nucleic acids. Nucleic acids are made up of nucleotides, so that is our monomer or a building block. And a nucleotide consists of a phosphate group. So phosphate group is a phosphorus atom that's at the center with oxygen surrounding it. Notice there's these little charges here with our phosphates. And then we have a sugar that is attached to the phosphate. And then there is one of 4 different nitrogenous bases. Okay, so you need to know this, that a nucleotide consists of a phosphate, sugar, and a base. Just kind of say that over and over again. Phosphate, sugar, base. Phosphate, sugar, base. Like before you go to bed at night, say phosphate, sugar, base. Right? You need to remember that that is the component, because we're going to once again spend more time talking about these molecules. So we mentioned that DNA and RNA are 2 types or 2 examples of nucleic acids. And one's called deoxyribonucleic acid and the other one's ribonucleic acid. So what are the differences between these two molecules? So here on the left we have DNA and right below it we have RNA. And if you just look at the image, you should see something right away. And that is the fact that DNA consists of 2 nucleotide strands. So this kind of blue backbone here, that's one strand of DNA. And then the other one spirals around it. And this is why we call DNA a double helix. RNA typically consists of just one nucleotide strand. All right, so it just has one and DNA has 2. And that's what they're showing in this image, but they've kind of flattened it out so that you can see the sugar, or excuse me, the phosphate, the sugar, and then the bases. Phosphate, sugar, base. Phosphate, sugar, base. And then down below it is phosphate, sugar, base right across from it to make up these 2. There's one strand and there's our second nucleotide strand. In RNA, we just have the phosphate, sugar, and the base. And this is just one nucleotide strand. If we look at the building block, the nucleotide, the phosphate, the sugar, the base, there's a slight difference in the sugar molecule between our DNA and our RNA. All right, so I'm going to erase that right there because I drew right across where you needed to look. So for DNA, notice we've got a OH here, a hydroxyl group, and then we have a hydrogen. And this is on the second and the third carbon. When we look at the RNA molecule, we still have that hydroxyl group there, the OH, but we have another OH right beside it. So it's a difference really of just one oxygen. All right, so this hydrogen here does not have an oxygen attached to it. This one does in RNA. And that's where we get our deoxy, kind of missing that oxygen, versus our ribo. Okay? Now, you need to know the 4 different nitrogenous bases, right? So this is where the nitrogenous base is attached. Notice there's a difference in how these 2 nitrogenous bases appear, and that's because there are different types of nitrogenous bases. There's only 4 different ones, but you need to remember all 4. So there's adenine, thymine, guanine, and cytosine. So that means we can make 4 different nucleotides because we would have a phosphate base and then an adenine attached, or a phosphate, I should have said phosphate sugar and adenine attached, or a phosphate sugar thymine attached, phosphate sugar guanine attached, or a phosphate sugar and cytosine attached. Okay, so there are 4 different nucleotides that can be made. Now the difference is in RNA, thymine is lacking. So an RNA molecule does not contain the nitrogenous base thymine. It's replaced by one that's called uracil, but we'll get into that later on. So there's still 4 different bases, nitrogenous bases for RNA. Okay, but start working on remembering some of this information so that when we come across it later on in the semester, it'll be a lot easier for you.